I'd like to welcome everyone to the American Cannabis, sponsored by Cannabis Tech. I'm your host, Ellis Smith, and today we're going to be speaking with Dr. Mitch Day on micropropagation of cannabis. Mitch, how are you doing today? Thanks for coming in.
I'm doing well. I appreciate the opportunity to talk about one of my favorite things, so thank you.
You bet. And so today we're going to talk about plant tissue culture, but also I want to give a little quick background on Mitch. And so his background is in plant tissue culture, data visualization, public outreach, conservation genomics, and bioinformatics. If you would, please give us a little understanding of what some of those things mean.
What are those things? Well, just I guess I'll just do it biographically in the order that my career kind of advanced. As an undergraduate, very early on, I got introduced to plant tissue culture as a method for laboratory research. I was working with some geneticists that were looking at mutations in flowering genes. And we needed a way to short-circuit the plant reproductive life cycle. Basically, these guys couldn't have sex because they had weird parts. And that's what it was about. I mean, how do you get around it? And, you know, I led a project to develop a method to short-circuit the plant life cycle, double up the genome without using sex so that you could see what those genes actually do. Pure basic research. It's an extremely interesting method, but that was just something I did as an undergrad. I moved on into the GIS field, which is basically Google Maps is really a GIS that everyone uses and doesn't even realize it. My public outreach work was related to GIS, where essentially we were working with large groups of business owners all up and down the Mississippi River. We wanted to do something cooperative to encourage economic development along that corridor, but we didn't know how to do it. So it was a sort of a stakeholder outreach effort where we used digital mapping resources to get ideas generated. And so we would intentionally make maps that were left unfinished and incomplete and take those to public meetings. Even though we had more data than we were presenting, the purpose of it was to stimulate discussion and to get people to fill in their own blanks. It was a very useful experience for that, because when you're dealing with technical subjects like we're dealing with today, you have to understand your audience and you have to engage them, otherwise they give you that look, which scientists get. I mean, we early in our careers we're excited with what we know, and we have to learn how to share that with as many people as possible if we're going to get any attention. Most recently in my career, I've been working in a field called conservation genomics, and essentially that's just applying DNA sequencing and analysis to figuring out how to save endangered species. In my case, particularly foxes and trout. Bioinformatics, just to explain that term for those that don't know it, it's really just computer science applied to really, really big data sets. So, you hear about data science all the time. It's a type of data science that involves working with data sets that might be terabytes in size. So anyway, that's a nutshell version of where I come from professionally and the things that interest me.
Pretty cool. Very interesting background, very diverse. I believe you also grew up farming as a kid, too, if you want to kind of touch on that.
Oh, yeah.
If
we're getting my life story. I was born in Boulder, and my parents fled Boulder because they're not crunchy. They're not. Most of my extended family's in Idaho, so we actually, growing up, had hydroponic greenhouses growing tomatoes and European cucumbers. Back in the day, the genetics of those European cucumbers were not stable, and so you'd have yard-long cucumbers that you couldn't sell because there's nothing to do with that except play baseball with green tomatoes you couldn't sell. So, yeah.
Interesting.
That's kind of where I come from.
Cool. So I love that you have a long back history. So
yeah.
I've known Mitch for over 3 years now, and he and I have kind of had an indirect working relationship, and I've really watched Mitch, get into the cannabis space and develop micropropagation. And it's cool to have him on here today, as I've been kicking tires with him for many years and have some things in the works, and just glad to have you here. And so today our objectives are really to inform, start a conversation, and collaborate and learn. And so let's jump in here. What is this micropropagation? Explain to us what this is. What are we talking about today?
Micropropagation is almost exactly what it sounds like. It's propagating a plant. Cloning, as we know in the cannabis industry, I don't think I need to explain that to this audience, why we do it and what it is. Micropropagation is just cloning, except that you use incredibly small pieces of tissue. It's just one of many methods that fall under the umbrella of plant tissue culture, which is a commonly used I guess, umbrella term, but micropropagation is just a precise term that describes a very specific method. You can see in the figure that I have up on this slide, you'll see it's a culture container, got a clear growing medium in it, and you'll see some little bits of green matter that have been encapsulated in that gel. Those are just the growing points that came from the plant, and they are ready to regrow.
How big is that in relative size? What is it, like a dime size?
If you're viewing this on about a 13-inch monitor on your laptop, that is just slightly larger than life size.
In scale.
Yeah.
Okay. So it's a very small piece of plant that you've, basically chiseled down. Is basically what you've done, correct? And we'll get into that in a little more detail.
Yeah, that's the what.
Okay. All right. Okay, let's explain this whole plant life cycle here and really kind of walk us through this process and kind of show us how this really makes sense.
Sure. Well, before we can get into the why, we start with the what. And this might be 101 for a lot of folks, but not everyone in attendance may be a plant biologist. So I just want to have a little common vocabulary so we can all know what we're talking about. This is a little diagram I put up that just shows the plant life cycle. You can get plants from seeds or you can get them from clones. And you can cut them. I just like this figure because it just shows how all things relate. And I like to keep track of information. It's part of my professional training. And so when you do that, diagrams like this is called an entity relationship diagram in the database design world. It's how you start when you're trying to figure out how you're going to keep track of a lot of information. And that's something that is important with micropropagation. Micropropagation is just propagating plants. Why are you propagating them, though, is the interesting question. And that's why I wanted to start with this, because it's into the why. And I hope by the end of the presentation, that's why I think this is a good way to begin.
Perfect. Okay, if you want to just continue on with just the plant anatomy here and just kind of walk us through with some of these visual aids here that you have for us.
Sure, yeah. This may be a familiar organism for people here on the right. It's a cannabis plant. It's a little bit stretched out, but I chose this image because it shows the features of the plant anatomy that I want to highlight. There are nodes, which are those areas there where the leaves are emerging from the plant stem. And the axil is just that little angle between the emerging leaf and the stem. And then the internode is just the space between nodes. And the growing tip of any of those branches that you see there is called the meristem. And the meristem is where the action is. That's the interesting part of a plant where all the growth and cell division is really happening. You'll see in the other figure, that's a figure that I borrowed from Wikimedia because it's a nice, clear image. It's actually a coleus, a common garden plant, but it has a nice structure that's very typical of all plants. You can see those arrows pointing to two areas where the cell walls, that's what those reddish zones are. There's a lot of cells packed in very, very tightly there. And those are called meristems. Those are the growing tip, and that little tip will eventually elongate, and those leaves that spin off from the side will eventually grow into full leaves. And if there's a meristem off to the side, like that one on the right, that may become a branch. And those are the parts that we're interested in because they're cells that know that they need to be growing. That's their mission, and that's what they're set up to do. They're the ones that make new cells. The other cells, they've got their mission and their fate in life is set, and they just can expand, but they can't really divide a whole lot. If you want to grow new plants, that's where you have to live.
All right, let's understand some of this terminology. We hear this term quite a bit, tissue culture, and I think even myself, I've kind of learned that for the type of processing that we're doing as far as propagating is really micropropagation on some of the stuff that I've been looking at. And so if you could just bring a little more clarity, because these terms are confusing,
Absolutely.
and really break down what the differences in these four different types of propagation are.
If you get lost in a web search and start searching these techniques, you'll see this information organized in a lot of different ways because it is, again, a complicated topic that involves a lot of different potential methods and applications. But I try to simplify it by just dividing the methods up into, according to how finely you chop up the plant material. So stem propagation, that's just another way of describing traditional cloning. I'm going to advance the slide to the next screen because I have a little table that has those same points of information with a little bit of additional information. So I'll use that to guide my discussion. Stem propagation is cloning, and why do you do it? Really, the only reason to do it is to propagate. You want more of what you have, and you have a pair of scissors. You chop off a stem, and you have another plant. Micropropagation, you're just taking it a little bit further. You get a better pair of scissors. You might use a razor blade, but you're getting down to the tip. And it's a small bit of tissue, but it's still quite visible and something you can handle without using magnification, but you would need tweezers.
And it requires a little bit more care at this point, correct? And food source and lighting. And
at this point, the plant is so damaged and cut down that the stem can't survive on its own. Cloning, even when you're doing stem propagation and you're cloning, you still have to baby the clone to a certain extent. It's not hard. You keep it moist and not swampy and give it a little bit of light.
That's it.
But it still requires more care than a seedling might. And it only gets more difficult to take care of these cultures as you move down on this table. In fact, this table is sort of cumulative. On my why do it column, the reason that you do stem propagation is for propagation, but that's also the reason you do any of the other methods. That's what I mean by that. So for micropropagation, you also would use that for large-scale propagation if you're in the wholesale nursery business, for instance. Meristem culture is essentially you're getting even finer. You might need a microscope or a dissecting scope, but you're still chopping things up mechanically or physically. This method is very well known in the commercial orchid industry as one of the ways they first figured out how to get viruses out of the orchids. It just involved micro dissection of the shoot tip of a valuable orchid and generate quite a lot of plants from that. So if you think back to that figure where I showed the meristem, essentially in orchids, the meristems are very, very densely packed. And so if you get a tiny one and chop it up, generate them very easily because that's already what they were programmed to do. Protoplast culture is where things get very, very interesting, and that's where you can't use any mechanical cutting anymore. You have to use chemistry. Dissolve the cell wall around cells, and you end up with a bunch of naked cells that then you can plate them out and regrow them. You can do all kinds of things with them, but in the end, after many more steps than in any of the other processes, you generate whole plants again.
Okay, wow. Okay, let's dive in a little bit deeper here and let's really understand and break this down some. Why micropropagate? Well, you kind of have some examples here on this methods compare, but really let's dive into a little bit more deeper and really understand this.
Yeah, why specifically would you want to use micropropagation? I'm focused on that today because it's the most accessible method. It's something that honestly you can use, I'd say not kitchen technology, but super kitchen technology. It's something that doesn't require a whole lot of laboratory stuff, a really good scale and a pressure cooker. You begin micropropagation. The one reason you might want to do it is you have fewer microbial hitchhikers. And what that means, well, you see this image there on the slide. That's a petri dish, and the little white smear that you see, that's an organism that I've isolated from the inside of many cannabis cultivars that I've tried to culture in the past. Now, I know they came from the inside because of the multiple times you sterilize the surface. Put that into a culture and you see something that grows, you know it didn't come from the surface because you eradicated everything that was living there. I was able to re-isolate this organism multiple times. We had it sequenced, and it's a bacillus that's very, very close to the bacillus people add as beneficials to their soils. This is a real thing. They're called endophytes, and endophytes are actually beneficial to plants in many cases. We don't know if this is an endophyte in cannabis. At least I don't. If someone does, that's a conversation I want to have because this is something that's curious. It was just a particular interesting one. But if you micropropagate under the right culture conditions, you can get rid of these kinds of hitchhikers, the ones on the surface and ones that are in the cells. The smaller the microbial hitchhiker, the more challenging the eradication. But bacteria tend to be.
Explain to me how, when you say clean, eradicate, how do you physically remove this bacteria or any other type from this, or even viruses? How do you do this? What is there a Kleenex or Clorox bleach? What are you using? And how is it?
Well, it's the philosophy behind it is the best way to defeat your enemies is to outlive them or outgrow them. So what you do is you do the sterilization that you can. Plants can survive being dipped in bleach as long as there's no cut surface there and you don't use too much bleach. So you can kill some things on the surface and get started. But really the final step that you've got to get right in order to eradicate things, you've got to encourage growth conditions that are so good for the plant that it can grow very, very quickly and basically entomb any remaining pathogens that the chemical treatment doesn't kill. You've got to basically entomb them in plant tissues. That's how plants work. They segregate and entomb their pathogens in tissue. They don't have an active immune system like people do. Basically just outgrow their pathogens. So you just help them along.
Interesting. Wow. And then so as we look at traditional cloning practices, we know it requires a lot of mother stock. It's very labor intensive. There's a lot of space that is in there that's required to really achieve a high volume. Micropropagation allows us to really streamline our labor force, really condense that space down into a higher volume of units. And so as we look at this, as our industry is evolving, comes down to your bottom line of your business when it hits a certain scale, and this makes more sense from a labor standpoint and even space allocation for how you're going to, just from those two components alone, but then you also look at the added value of contaminated-free genetics and all these things. So explain that a little bit more on why this is so important, and as we scale and our industry is growing, we're going to be shifting and changing, and the way we're doing things now are going to be very different the next 5 or 10 years as this technology becomes more readily available and people buy into it.
Right, it is a matter of scale. This implementing micropropagation is a complicated operational decision. There's a lot of different factors. There's a certain point in your operation, though, when it becomes cost-effective. You have a large enough production goal, or you have genetics that are precious enough or valuable enough to take the care to. That's when this becomes a very useful method. And we don't have to invent anything in the cannabis industry. We have a long history of these kinds of methods being used in crop improvement and in the horticulture industry for the better part of a century now. We're just learning from past on this. We've got some things you have to do to customize and research specific for this species, but the general principles are well known. One of the first reasons that we can make the claim that it's more labor efficient is honestly just the amount of care and the number of touches that you have to have on your sample. It takes a bit of care to prep a plant and harvest the material, but it's all done in a very small, compact space, a single worker with fast hands, and quite a large number. And once the cultures are sitting on the shelf and you have a properly designed facility and you have your sanitation and your aseptic methods worked out, you very rarely have to visit the cultures for long periods of time. So they can sit and do their growing, and there are interesting challenges when it comes to designing these shelving systems. That's it. Invest in the capital and the labor saving of find a few good, efficient.
The ROI will come, one, from your labor, but also, too, just your improvement in your crops and just consistency, from starting with clean material. I think that's added value that I think we're going to see more and more folks dive into.
Yeah, I expect this method. I mean, we already know there are people entering into the industry that are interested in, that have been applying this method. There's been a lot of discussion about it on the various online groups where the serious folks meet and talk about this stuff. It's here, but the trick is getting it right and using it for the right reasons in the right place at the right scale.
And then kind of explain why it's good that these cultures can be slow and grown and kept for long periods with minimal care. I know the answer, but I want you to tell our audience just the value behind this type of methodology without really having to have mother stock,
right,
in that mindset.
Well, genetics are valuable. I think that's it's a constant topic of discussion in this industry about, how do we treat our genetics? How do we make them valuable? How do we reward the people who make better genetics? Well, you can't get any value from your genetics if you've lost them. And mother plants are large. They're relatively expensive to keep. You can keep a large number of things on the shelf that you only have to look at occasionally. You have a larger genetic library to draw from. That makes you a better reader. Got more to work with.
Well, this is the meat of, I think, our conversation today, and I think people are going to be excited to really understand this particular part of this, of our talk today, is how do we micropropagate? Where, how does this process start? Where does, and how does it end, and what are the steps in between? And it's something that, once I understood it, I was told many different urban myths on, tissue culture and what it really was, and now, once I discovered what I was told and what I learned, it was two different things, and so I think this is going to be exciting for our audience to really kind of dive into this component here. So yeah, let's go through these slides and really break down, how do you do this?
All right. Well, yeah, I've given you a broad overview of all of the potential methods, but we're just going to focus on the one, micropropagation, the one that I think is the most accessible to our audience, and one that people could consider beginning using right away. Information you can use, I hope. You got to get started with the mother plant. You have to. You can use pruning techniques to prune the mother plant to produce as many of those fast-growing meristems as you can. There are some cannabis terms out there. I think florabunding was, Ed Rosenthal was authored that term, or he coined that term for pinching up your plant in a certain way to get as many things as you could for cloning. Any kind of a method like that just pinches that plant up. So there's lots of good growing tips. Is how you get started. It's the right kind of material. It needs to be thin and non-woody, and if it's in an active state of growth when you put it into a culture container and you do it right, it'll remain in an active state. That's the nutshell of it. How you prepare that starting material is you harvest it, you cut it, bleach it, you rinse it under very, very clean conditions. And I'll talk a little bit more about that later, so if there are questions about what I mean about that, that's something I'm going to cover a little bit later, but I think it warrants a separate discussion of its own, so I'm going to put a pin in that. All right?
Sure.
All right.
Real quick, I want to answer this question as we're kind of getting to the storage part when you clean, chop, and coddle and getting to the storage here.
Absolutely.
And maybe a little premature, but I want to get this out of the way as one of our listeners has had this up for a few minutes. His question is PTC a better storage method in seeds for long-term storage if the cultures are kept in a controlled environment?
That's a good question. It's a different kind of storage. Seeds require even less care, cool temperatures, relatively, controlled humidity, and you can get germination decades from a large, sample of seeds. So they're very different. The reasons that you would keep seeds are different than you would keep cultures. You've got a genotype that you love, and you don't know why you love it, or you don't know the genetic reason that you love it. You want to keep a clone. You want to keep it in a plant tissue culture state. You might want to propagate that for commercial production. If you're a visionary and you're a plant breeder and you've got a vision about what you want, and you know some male parents that might want to get together with your female plants, you've got seeds that you can keep, and you understand that are an entirely different kind of product. The future is seeds. Micropropagation is a method, but plants take real plants from.
Yep.
So it comes down to seeds. Ultimately, you use micropropagation as part of a bigger effort to make better seeds.
Okay. I think we don't always have the luxury of seed with some of these varieties. Sometimes we get clone-only varieties, and that may be your only outlet of storage. And so I think that's something to also play into the conversation.
Oh, absolutely true. Some cultivars that I've worked with and brought to seed are extremely poor seed producers. They were never selected for that, and they're extremely poor seed producers. That's a genetic flaw in their background. They are a wonderful producer, or it may be the reason that they're a wonderful producer. Find out.
That's it.
So,
That's it.
yeah.
All right, explain to us these steps and variations in the process, and the sugar and the nutrients required, and really just as you get these things cleaned and chopped, that coddling. What is that coddling?
Well, there are many variations of this process, so I'll be a little bit general, but there is one universal requirement. Plants, any living organism, essentially, has to have sugar to live. You eat food, but it's ultimately turned into sugar. Plants photosynthesize to make sugars. When you remove all their leaves, you remove their ability to do that. So for a while they're small and in this encapsulated state that you see in this figure that I've reused here, you've got to feed them some external carbon. That's in the form of sugar. That's useful. It's a good, efficient food source. It gets them started. They can unfurl their own leaves and begin making their own food. But at some point, the sugar becomes a problem of its own, and I'll talk about that in some future steps. But the sugar is an opportunity and a challenge. You've got to feed it to them. That is a universal requirement for this method, regardless of your variations that you use. Now, how do you coddle them? How do you chop them? The method that I've implemented in this particular figure, you see those growing tips. They look like they're in some sort of a bead of their own, and so they are. This was a two-step process. The gel material that fills most of the culture container is geldan, or gelrite. It's a seaweed-based growing medium common in microbiology and in plant tissue culture. But the growing tips were encapsulated in a separate process in a related material called alginate, which sets up differently. You don't melt it, you use chemistry to get them to set up. You take those freshly cut tips, and before they can dry out and get stressed, you quickly encapsulate them by coating them with alginate, dropping them into a solution that treats them very gently, but gives the calcium that's needed for that little setup. It's basically a cradle or a womb for the growing tip to survive.
I was about to use that analogy, like a womb for it, basically.
Exactly. And the chemical composition of both of those gels in that picture are kept as similar as possible because they will be eventually anyway by diffusion there. Treat them gently, and if you do it right, you will see growth cracking out of those beads, sometimes honestly within 48 hours or less.
Oh, wow.
If you're doing it right and you have your conditions right, including the quality of your starting material,
that's great.
Your growing conditions, and that does vary cultivar by cultivar too. Some cultivars love being chopped up.
Do you create these recipes for these embryos, I'll call them, or these little nutrient sacks, I guess, or do you purchase them that's already pre-made for this?
Well, it's a little bit of both. I mean, there are some very common methods, some very common nutrient solutions that are really just part of the public domain. They've been known for years. They were proven. There's a salt mixture that was developed in tobacco, because that was used as a research plant for very, very many years. And so this was developed in academic settings, and so it's a matter of public knowledge. But then there's all the other additives, sugar, the antimicrobials that you have to add. Sometimes some methods use plant growth regulators, those sorts of things. That's where the mixing comes. Generally, the standard salt mixtures, there are so many different varieties of them that have been. There's not much invention there, although a large-scale operation, there is tweaking. Certain minor constituents might want to tweak those to optimize the process. But that's something that happens as you go along and as you scale. The generic formulas, those are well known and can be found by anyone with some Google skills.
Okay, now at this point in the process, this is where you could store long-term genetics and
right.
Slow the growth down. As we go back to a few slides, we're talking about slowing it down. Is this at the stage you would do this at for long-term storage? One, because it's such small plantlets, and two, you can put a lot in this space.
And this is just one of the methods for storing genetic material long-term. I'm just focusing on the micropropagation method, again, because it's the most accessible. I'm trying to get this kind of information out to a big audience. The method that you can use is the one you should use.
Okay.
All you really need to do to slow them down at this state is just lower the temperature and dim the lights. they just slow down. They still keep growing. Still have to look at them once in a while, but they just hum along and operate at a lower temperature. Chemical reactions in general just go slower at lower temperatures, and so growth just has to go slower because of physics.
Well, this leads into a great question from someone who's saying, how long can cannabis stock be stored in tissue culture without degradation? I think this kind of leads into this.
Well, that's a question that comes up a lot, and I'm assuming that the questioner, please chime in if I'm misinterpreting your question, but that you mean genetic degradation. I'm assuming that's the background, because that's usually
sure. Or even with the, I guess that can be looked at in two ways. Yes, one, genetic degradation, but two, could the plant actually degrade, like full-on decompose, and actually decompose, I guess is the word I would probably use.
Well, absolutely, yes. If you're storing plants this way, you are still going to have to occasionally do a process that I call subculturing, the common method where essentially you would just take the same material, chop it up, and coddle it again. So indefinitely is the answer. Now when it comes to genetic degradation, that's a different story. Mutation in plants stored this way is extremely rare. Not really disrupting the plant at a cellular level. You're not doing anything to cause mutation.
Great.
Now, but there is a question mark there, though, because of the relative infancy of cannabis genetic research. There may be instances of it, but if you look at the literature for all other crop and horticultural and research species that have been subjected to that kind of testing, chromosomal or genetic mutations using this particular method are very, very, very rarely.
Okay. Well, now that we've got these the plantlets going, and we are finally getting some growth, and you start to see some leaves, what's the next step here? You got the slide as deflask and harden, and I think some of us may know that term hardening from either our seed starting or even our cloning, and so how does that work?
Yeah. Anyone that grows in a greenhouse knows the term hardening, because you just like us, treat us too nicely, we get a little soft and can't handle the real world.
I love that analogy.
It's even more apt here, because you see that little culture container. That thing is completely protected from the outside environment. There's even a little filter on the top of that culture container that keeps anything from getting in that's bacterial. The only thing you can get in and out is air and a little bit of water vapor. So it's living in a bubble. And there's all that sugar on it. You bring that out into the real world, leave all that sugar on it, that's just a feast for any pathogen microbe that lands on it that's in the air. So again, clean conditions and gentle introduction to the real world. Now, getting that sugar off is a major process challenge that's a very significant one, and probably one that will determine your success or failure at doing this cost-effective and efficient way. And, generally it involves just manual work.
Really?
Yeah.
Just that easy?
Yeah, it is. It's just something you have to do. You just have to do it careful and make sure you've got good processes and checks to your processes, some good quality control.
Well, it sounds like it's at this stage in the plant now, you're following the same protocol, basically, of a clone coming out of a clone dome, and
absolutely,
trying to monitor it in that same perspective.
Those little plants that you see in the lower figure, those have just come out of the flask. They've been in a dome for perhaps 2 or 3 days, and they're tough enough now they can handle 70% humidity. But they are only about 2 inches tall. So that's only one difference. Your transplanting process might be a little differently because you're working with material that's a bit smaller.
A little bit shorter.
Yeah, so you got to take better care of it. Different care of it once you're getting it to soil. But yeah, once you're to this point, got root growth, dealing with the plant.
At this point now, you would never know this was done from micropropagation at this point if no one ever showed you what happened in the back end and just presented this to you. You would just think, oh, wow, that's a small clone.
Yeah.
It's got a lot of foliage, looks great, healthy. It has a little funky little clear little food source, I guess. Is that what that is?
Yeah, that's honestly just there's a balance you have to strike. When you yank these things out of the auger, you can damage their roots. But they work so hard to grow. But if you can get the timing right, they don't have a whole lot of roots, but they're just ready to blow up.
Cool.
On root growth. So this was a variation on a method that we tried quite a while ago that just used that same gel material to hold them in place. It was very successful in the pilot that we tried. We've moved on to other methods. But it was a very useful method.
Okay. Source meristems from cultures. Explain this more to me and what this means. I personally want to just understand a little more in detail.
Sure, absolutely. Well, we've spent a lot of time talking bad about mother plants, and that's because they're expensive. I mean, we all treat our mothers well, and but that costs money. And ultimately, if you want to do this process on a continuous basis, you can start sourcing your meristems from within those culture containers. I showed this particular picture because it's actually 5 different plants jammed in there, just like in the earlier slides I showed, but these are more advanced in growth. They've had a chance to even branch. There are probably 20 meristems in that container, and if you grew it a little bit more, you'd have even more. Pop that open, get out your tiny scissors, and just stems you would off a 2-gallon mother.
Now, isn't that interesting? Now, that just gives you some perspective, and that's why I want to ask you this, because I've never heard you explain this to me before. I haven't heard anybody explain this to me before, and this is just so interesting to see how we can now really maximize, once again, in a large commercial setting, when you've got 2,000, 3,000, 4,000 square foot allocated to your vegetative space and your mother space, and call it another 500 to 1,000 square foot for your cloning. Imagine how you can turn a lot of that square footage into more revenue producing square footage for you, and really just using your space more wisely. Mm-hmm. And so, once again, when you hit that certain scale, it makes sense to really look at bringing this in, because it impacts your bottom line and can really change the way you design and build your facilities from day one. And I think that is what's exciting about this, is it's really a game changer at a certain scale in my eyes.
Right. It's more of an operational challenge to drop it in from a facility standpoint. It can be this technique can be dropped into existing facilities. That's relatively easy, but we all know how changing operational processes is. That's where the real challenge is integrating this into your workflow.
Well, I'm still fascinated by this, because you're showing me a visual of 3 inches tall by 3 inches wide. That's 9 inches. 9 square inches, you're able to pull out 20 meristems, and anybody knows that's a top on a plant. So you can imagine, in order to grow a plant that has 9 tops for you to harvest clones off of, think about that. Mm-hmm. It just puts perspective for me, and I'm still just, I'm in awe.
And you can get a lot more meristems than
sure.
that. If you're harvesting something like this in a production scale, that thing looks like a tangled box of salad. It's just
awesome.
A jungle.
Awesome. Yeah. So you can definitely get probably 100 in that space.
Yep. Once you get your conditions optimized.
So this is the true benefit behind the technology, and I think for me this was just eye-opening to really see just that ability in 9 square inches, what you can really pull out from a production standpoint.
Right.
And I think this should just really hit home to our audience of just how neat this is. And really, as we look at scaling, this is really where our industry is going to go. It already is. So how do you build one of these things? What does it require? What goes into this? Does it require serious protocols and clean protocols with decontamination chambers to go in and out of, or can you get away with just alcohol and rubber gloves and a hairnet and maybe a little vent fan to help pull air out? Or how sophisticated does it have to be? And I'm sure you can tell me the sky's the limit, or as you show me the picture here, you can show me how simplified this little small workspace is that you were very productive in. And so.
Absolutely, yeah. It's somewhere in between. If anyone's a Michael Crichton fan, remember the Andromeda strain and the disinfection procedure they had to go through?
Exactly.
That's an old movie nerd reference, but it's not necessary to go that extreme. It's not necessary to use even, honestly, I would say laboratory. You just have to have a very clean environment that's segregated from your main production facility. The images that you're seeing on this slide, actually I want to shout out the people that hosted me there. This is in one of Green Dream Health Services growing facilities. They hosted us there, and I want to thank them for that, and Shift Cannabis, their business partners. They were gracious enough to give us 100 square feet, and we made very good use of it. Always be grateful for their mentorship and hosting us. You see some pictures there of the ceiling, that middle facility there. That's just a HEPA filter that's on the end of a duct, and it recirculates most of the air in the room and then draws in. It was about 10% was the amount of air that was drawn in from the outside, and then the rest of the fan's blower capacity was just recirculating air in the room already through that filter, through that HEPA filter. And you'll notice on the panorama there, that's a little bit skewed. So if you're having trouble orienting, I'm basically in a 10 by 10 room standing in the corner, and I took a panorama so it looks really spacious.
To enhance it.
Yeah, there was no twirling or dancing going on in here, though. It was actually a very compact space.
So you can just see, 100 square feet, 10 by 10.
Mm-hmm. The only exception to that footprint is that little sterilizer that I've got in the upper image. That had to be in a different room purely for facility requirements. But that's basically just a fancy pressure cooker that is very efficient and safe and faster than a pressure cooker and more consistent for sterilizing the medium and getting everything clean. You can do it that way because you seal your stuff in a jar, walk down the hall, drop it in the sterilizer, bring it back while it's still sealed in the room. Now, going back to the filtering, actually, I wanted to point that out because I didn't want to get detracted from that, because it's an important consideration. To see the door is open at the moment. When this room is in full operation, that door is closed, and there's just a vent at the bottom of the door. You've created what's called a positive pressure work zone. And what that means is that fan on the HEPA filter unit is pulling in some air from outside, and it's got nowhere else to go except for out through that grate in the door. So there's a positive flow of air outside of the room. It's all clean that's coming out, so it basically sweeps microbes and contaminants. And right down that hallway, there are several active grow rooms, flower and veg. And we did not have problems with contamination. The process we did.
Awesome. And so you see the workspace, you see what it takes to get the environmental controls where it's needed. Now the operational requirements, how do we manage this? How do we keep everything in order? How do we not mix up all of these tiny little boxes with all of these different little plantlets they all look identical? How do you manage that?
Well, when I often tell people that this process isn't magic, but actually this is where the magic is. You have to have a good laboratory manager. You have to have a good process. Basically, any operational competence that you have to maintain a large-scale facility, you've got to apply it here. You have to have record-keeping and labeling practices. You've got to have inspection schedules. Time to sit and baby 5 or 6 individual cultures. You've got to deal with, ideally, thousands if you're doing it right. How do you keep track of them? That's a big challenge, and part of the conversation that I want to start with people. There's some real opportunities there, figuring out creative ways to label these, to keep track of them. There's all kinds of companies that are entering into the cannabis space that have all kinds of interesting technologies for tracking things. This is definitely an area where those are the most critical, most important. A switched tag in this context, if you're using this for breeding decisions, label mistakes can have you don't see.
That is true, isn't that? So interesting. Huh. I got a few questions here of some folks that have hit in there, kind of go back on a few questions. Let's see if we can hit on some of these here before we kind of wrap this thing up on the last slide that I know we want to kind of touch on and give us some time to explain on. Is there any more of a concern about genetic mutations with protoplast culture?
In general, yes.
Because it's now broken down and so vulnerable, I guess I'll use that term. Is that why?
Yeah, that's the general explanation for the mechanism for why it might happen. I'm just really referring to the literature that has looked at this question across all kinds of different, a little bit more of a problem. However, I want to emphasize that in general, a lot of major concerns. Plants have been on this planet quite a long time, longer than us, and they know how to protect their genetic information. They have to take care of it. So yes, mutation is something you have to look for, but scale of things, it's a lower level concern that I think really it's about doing this and phenotyping your plants, understanding what they. You'll notice there are genetic tests and things. Genetic integrity. That was a major part of my conservation genomics research that I talked about earlier. We were looking for levels of genetic diversity in whole populations, but genetic diversity.
This person is asking, with cloning, we know that we always are going to get the same plants. With micropropagation, it's the same, or can we have a problem with the genetics? And as I want to explain to them, and you can explain in detail more, micropropagation is just basically traditional cloning taken down to a little smaller part of the plant.
Exactly.
Right? So it's no different than a regular cloning process. So nothing changes, right?
Not when you're doing this method. I mean, this is something that we need to look at in this species. But we have so much information drawn from other crop species that this shouldn't be something that keeps. It's a minor part of your operation. It's something to count of, but it shouldn't be a reason that you don't.
Okay. Here's another question. Can you use UV sterilization over an autoclave?
UV sterilization works in many contexts. However, it depends on the chemistry of what you're sterilizing. UV is a good sterilizing agent because it attacks molecules and breaks chemical bonds. So if you've got something in your medium that is sensitive to UV and has a particular bond structure, it may be disrupted or chemically altered by the UV. However, the same is true of heat. If you heat your stuff, and you may get byproducts. So you just have to optimize your process. So the answer is, in general, you might be able to do it, but it may cause problems with some of your constituents in your. But the same can be said of heat, but those techniques are well known and solved through proper formulation. You would have to do the same.
What are the top 3 errors new operations make starting micropropagation?
Underestimating the amount of time it takes to bulk up your stock. That's the primary error. It takes quite a while. Your processes have to be ironed out. You've got to have a good, stable operation, and you've got to let the people work, bulk up their stock, and it doesn't take forever. But I think it's not. So make sure you understand the amount that it'll take. It also changes your production schedule because the time from cut soil in micropropagation is much longer. So build up a large mass of cultures in that time. You've got to build that backlog, allow those to bulk. Honestly, that's the first and second one. It's that important, and it's that easy to get wrong. It's just a production challenge, though. You have to project management.
I mean, you need to have that process on a perpetual cycle, just like you do your vegetative and your flowering stock, and so it's no different. And just understanding that timeline. Here's a question that came in, and see if we can try to answer this: how to reduce cost to scale micropropagation?
Count your steps and stop taking as many steps. Good process control, good Kaizen. Use your lean manufacturing practices. Study your processes. Continuously refine them. Labor is your big cost. Material costs are relatively fixed. There's some opportunity for economizing on your material costs, but your labor costs present.
Like the Kaizen reference, I was about to make that. I mean, constant improvement, always looking at process flow and making improvements and never being satisfied. And
right.
so I think that's a big component.
And that means, that key hires are important. Record-keeping processes are important. They're all part of a whole ecosystem. It has to fire on all.
Here's something we didn't answer, or we didn't touch on. I think this is a great question. How long on average does it take from putting an explant into culture to establishing it into veg? And I think that's always that big question. And so, what's our answer here for our audience?
It's highly variable based on cultivar. I did mention that earlier. I think that really liked being treated poorly this way, and they grow. And then others that are otherwise very robust growers in production were very reluctant to grow under these conditions. A range 8 to 14 weeks.
Okay.
Yeah.
Okay.
To the point where you're ready to.
Okay. Good.
You can probably do it faster. There's a lot of optimizations that can happen there, but that's what we've been able to do.
And as I mentioned before, as you see, I mean, that's quite a long time. It's 3 weeks or 3 months plus. But in the big scheme of things, once you get on a perpetual cycle, you don't have to wait 3 months every time. You'll get it to where you'll understand you need how many units a month, and you'll get in that cycle. And sure, getting up to that cycle will take time. This is where another question comes in. How long does it take to get to a commercial level of production? And so as I was trying to explain, and I'll let Mitch explain more, you've got to get it to scale. It's going to take time, especially perpetually.
Yeah, really the answer to the question is just understand the frustrating and slow part of an exponential growth curve. Any exponential growth curve, you start with a small amount of material, start dividing things, and you plot that on a chart. There's an area there where things are relatively flat. Keep it going, and you don't disrupt that continuous round of doubling after doubling. It ramps up very, very quickly. So really do allow months. But
months, yeah.
Not years.
Yeah, and I can tell you for some of the projects that we have been speccing this into the business model, it starts out with traditional cloning. You've got to do that to get going, and we are looking to scale into this, and it's over a 9 month to a year time to really replace our traditional propagation from cloning into this methodology. And so just think about that as our audience is looking to possibly do this, that it will take some time, but when you finally make that switch, you'll really see the impact.
I want to end, if we may, at least on the discussion. I definitely want to have as many questions from the audience as you have, so I'm not wrapping up here, but I do want to kind of throw something new, sort of end on a different note.
Let's do this.
Yeah, let's talk about the bigger vision. I mean, why are you doing this? This is one tool in your toolkit. And in reference to an earlier questioner that was asking about the 3 big mistakes, the third one that I don't think I mentioned is overestimating any method. Every method has a utility. Every method has a place. Micropropagation is just one method. Applications. Why are you going to do it? You've got a toolkit. What are you building? We're building better genetics. We're building better plants. That's the goal, and that's the future of this industry. I put a little graph on this last slide, a little bit of explanation. The y-axis there is bushels per acre of corn yield, and the x-axis is the year 1866 to 2017. Those are corn yields in the U.S. Look at what happened around 1940. You have to look at it very long to see the trend. What changed? People finally caught on to the idea of using double-cross hybrids in corn. Lo and behold, yields have continued to increase for about a century now. That's a long time, and that's a lot of improvement. A lot of other things have improved at the same time over the 20th century in corn, but if you break it down, and many people have, there's a lot of good research on this, about half or more of those yield increases that you see on that chart are due solely to improvement. Everything else that people have tried, fertilization, soil treatments, pesticides, spraying, whatever you do, that's the other half. So if you want the low-hanging fruit returns, you've got to be improving your genetics. I wanted to introduce that as a point of discussion aspects of this method and others that are entering the industry now, really, how do you keep your and micropropagation is appealing to scientists stuff around. Get a lot of data off of this stuff while you're making some money on it and running a.
Well, I just love this slide. Every time I look at it, it just makes me giggle, and you made a really good reference about how when the trend happened, probably half of the increase of yield was due to genetics, and the rest being all the other components from nutrients to all the other aspects. And I feel like that is where our industry is currently now. I feel like we've got a lot of great operators who have cracked what I call, you can grow the 3 a light. I'll use that metric, and now people are growing 4 a light. And I'm knowing guys that are now pushing the 5 and 6 pound a light in, call it 20 square feet or 25 square feet with 1,000-watt lights. They're doing it with nutrients and pruning and training techniques, but they haven't tapped into the genetic aspect here in the breeding side. And I think if we're already seeing leaps and bounds now in our industry with these types of yields over the last 3 or 4 years, imagine what we're going to see here in the future with the proper breeding to really go after yield, but also cannabinoids and terpenes and targeting specific elements and there's disease resistance,
ease of harvest,
all of these things, leaf to calyx ratio, these types of things, so that we don't have to trim it. You can just harvest it and it's ready to go and just try it. And so this is exciting.
If I can point something out in this chart that I think is a useful historical lesson for the industry too, the idea of creating hybrid corn is much older than 1940. It was honestly a method that was starting to come out of the research universities in the teens. But it took that long for enough people to try it and enough data to encourage mass adoption of these like double-cross hybrids. So there's that lag time. I don't think we can count on 20 years in this industry for lag time. I hope not. But the lesson is there. Breeding is a long-term investment, but it's where the long-term gains are as well.
I think this is where it goes into, creating something. I don't hate to use the word co-op, but really as our industry as a whole, making this a community outreach type program where we've got to get all of the operators involved in this to really participate in the breeding and the trialing, I guess is what I really want to use, because a lot of the varieties that are being bred right now aren't stable. They would never pass a seed certification standard ever in traditional horticulture, and I think this is where we're going to see our industry just grow by leaps and bounds as we start to make these moves.
Absolutely. This is where it has to go, and this is where it is. There are 2 different ways to do this. You can do it close to the soil, or you can do it big and bad. There are models for both, and when I say big and bad, I don't mean bad in that sense. I mean really the sense that you can be secretive, but that's expensive because you've got to do everything yourself. If you're a smaller operator, there are ways to become profitable in this industry and to participate in plant breeding, but you've got to focus on a more cooperative model. The secret to me in this industry is finding the right kind of deal structure where you can feel protected that your investment, your work, creating better genetics, protected somehow. There are plenty of new entrants into trying to address aspects of that problem, and definitely welcome them. They're all part of the ecosystem. All have to build. There are room for smaller players, but they've got to be smart, and they've got to figure out how to play with this game. I don't think there's anything about the general future of the industry that comes to being the smaller players that can play long-term from a production and breeding standpoint. Those guys, they're going to be the ones that are genetically sophisticated.
That's it. So. Good sustainable business model.
Mm-hmm.
Anything else you want to add on that? I know that's a very key part we wanted to really throw in here at the end.
No, except I invite people to reach out to us because I'm really serious about this being a conversation. The secretiveness, got to protect your secret sauce, but there's a lot of sauce that isn't secret. Let's talk about that.
I like it. All right, before we wrap this up, then I got a few more questions, and I think we're coming on the top of the hour here, and we'll just wrap this up. So Do you see a business opportunity to replace traditional clones and ship cultures only? And I can say yes. I can say that Mitch and I are looking into going down this pathway together to start businesses in different markets offering this service. And it goes big and beyond that, but I'll just kind of leave it at that. But yes, we definitely would agree with that. Wouldn't you say, Mitch, that this is definitely an opportunity to replace traditional clones?
Oh, absolutely. Yeah, and for transport, it's not a technical problem, it's regulatory. So yes.
100%.
You can do it. Just fill your forms out correctly.
And this is, maybe you can help me on this one as well. As I know the cost. Let's see what this says here. Do you have a rough idea of the production cost per clone that can be achieved with micropropagation? I can tell you to cut a clone, if you look at your cost from a labor standpoint and all of the inputs from your medium, the 14 days of power, all that, you're in about $2.30, give or take, call it 30 or 40 cents. And so to scale that same unit, what will that cost be down to?
It's going to be multiples less once you have your processes optimized. It's definitely competitive very quickly at cost. And from your experience in the cut flower industry, Ellis, you know that $2.30.
100%. So yeah, I mean, at the end of the day, yes, it's definitely a lot more cost effective. I'm assuming below 50 cents a unit, probably way less than that, just with the volume and scale. And so.
And honestly, that's the differentiator, is the people who have their operations leaning up to the point where that'll be a very good long-term advantage.
Well, I want to wrap this up. Just know this will be available online, and you will be able to access this usually within a few days. And so we just want to thank our audience for tuning in today. I want to thank Dr. Mitch Day for coming in today and talking about micropropagation. So thank you.
Thank you. It's been fun. I hope that it was entertaining, and I hope that there weren't any glassy eyes.
Well, this is a great topic that I have a lot of passion about and interest in myself.
Well, my eyes are lighting up. This is what I like to do. So hopefully I've reached my audience out here. So thanks everybody for attending.
I had fun today. It was great. I want to thank everybody for tuning in once again. I'm Ellis Smith. Thanks for tuning into the American Cannabis, and thanks our sponsor, Cannabis Tech. Everyone have a great day, and see you next month.